Good evening. Welcome to the session of NPTEL Cell Biology Cellular Organization, Division and Processes. This course is being taught by Professor Shikha Laloria, who is a professor in Biochemistry Department of IISc Bangalore. I'm one of the PMRF TAs of this course. I'm Smriti Sundar, a PhD student in IISc Bangalore. There'll be weekly discussion sessions thrice a week, one on Saturday, one on Monday, and one on Tuesday. This session is mainly meant for doubt clearing sessions and discussing the basic concepts that have been taught in this week. So, you can see here, it's a Petri dish containing different types of colonies of bacteria or fungi. These colonies are what is visible to our naked eye because bacteria and fungi are often microscopic creatures and you cannot often see them. If you can see here in this dish, there are different type of colonies of different colors, different shapes, different sizes, and often these are a characteristic of a species. They may be smooth, they may be wrinkled, may have a dry appearance, they may have a mucoid appearance, and they may have different colors growing on different uh, plates with different nutrient sources. But essentially, what they are made up of is millions and millions of cells put together. We have seen in the previous sessions that mitotic cell division is occurring in these cells, and it consists of different phases which have been tightly regulated by a large number of phosphatases, kinases, and cyclin proteins that have this cyclin expression levels in different stages of cell cycle that gives it its specificity. So these stages typically progress through a G1 phase, wherein the cell is getting ready for the DNA synthesis, and S phase, where the DNA is actively synthesized into two copies, the G2 phase, wherein the cell is getting ready for mitosis or cell division. It typically grows in size and it's synthesizing all the necessary proteins that is required for cell division. And ultimately has this dramatic phase known as mitosis, wherein the cell divides into two and there is equal distribution of the contents into the two daughter cells. And essentially, this is the reason that bacterial cells are able to form such large colonies in such short amount of time. So this is a very interesting question, wherein if a bacterial cell divides every minute and takes 60 minutes to produce a visible colony, how long will it take to produce half that amount? Now, considering a visible colony is around millions of cells, what would be the amount of time that is required to produce half the amount of cells? Now, because this in the question it said that the cell divides every 1 minute, half the cells, if they divide just once more, then it will produce the number of cells that is required for the colony. Hence, if it takes the bacterial cell 60 minutes to produce a visible colony, in fact, it takes it only 59 minutes to produce half that amount. Now, imagine there is a larger organism, such as a human, which will be capable of reproducing only every 30 years or so. It will take long time to produce a million cells. Now, the reason bacteria is able to achieve such fast division is because every colony that we see over here is a single clone, which means that all of these cells have the same genetic material. So all the cell needs to do is to replicate its DNA and divide. And there is no genetic diversity that is generated, no fusion of different gametes that is required. And that is why it is able to achieve such fast division over such short amount of time. However, for larger organisms like a human or a higher eukaryote, sexual reproduction comes into picture, and here it requires a fusion of a female and a male chromosome. This fusion requires that the DNA content in these cells are halved, so that upon fusion, it will still retain the original DNA amount. If this is not done, if the DNA content is not halved, with every fusion, there will be double in the amount of chromosomes present in the cell, and this will lead to catastrophic amounts very quickly. So, meiosis, which is the event that results in the halving of the DNA content is the key to successful sexual reproduction. So herein a depiction of the comparison between meiosis and mitosis. In mitosis, we've seen that the first phase, which is DNA replication, is followed quickly by the mitotic phase, resulting in two diploid daughter cells. However, in meiosis, if the parent is a diploid, that is, it has two copies of every chromosome, then first DNA replication happens, followed by pairing and crossing over of the duplicated homologs. And then the homologs pair with this pair up. First, they line up on the spindle, and then they are separated. The homologs are separated into two different daughter cells. And this is the first cell division, resulting in two daughter cells known as meiosis 1. And then later, each homolog present in the cell again lines up at the center of the cell, and now the sister chromatids are separated into two daughter cells, ultimately resulting in four daughter cells with a haploid condition. So things can go wrong very quickly in meiosis because it's such a complicated process. Because initially, already if the cell has 2N chromosomes, there is a stage after DNA replication over here, wherein the cell is having 4N chromosomes. This stage has such large amount of DNA content within it that proper segregation, equal number of chromosomes going to each daughter cell, is a very complicated process and needs to be highly regulated. Now, assumingly that this is the initial cell, it has 4 chromosomes. It has replicated, so each chromosome has a sister chromatid associated along with it, as we can see here. This needs to be aligned at the center of the cell. Now, supposing there is a mistake in this step, this mistake in meiosis is often termed as non-disjunction, which means that the chromosome is not equally divided into the two daughter cells. Say there is a non-disjunction early on in the meiosis 1 in itself. Then what happens is, actually in meiosis 1, what should have happened? One homolog should have gone here and the other homolog should have gone here. But because of a non-disjunction, what happens is both the homologs of this chromosome goes to one cell, one of the daughter cell, whereas the other daughter cell does not get any homologs. So this again, when it undergoes meiosis 2, what happens is the 4 daughter cells that is formed, 2 of them have an extra chromosome present in them, and 2 of them have a lesser number of chromosomes present in them. So herein, actually, because there are 4 chromosomes, what should have happened is 2 should have gone to one daughter cell and 2 should have gone to the other daughter cell in the end of meiosis 1. But here we can see that 3-1 has gone to the 2 daughter cells respectively, because of which this abnormal condition has been found. This can also happen in the second cycle of meiosis, which is meiosis 2. The same non-disjunction did not happen in meiosis 1. The chromosomes were segregated properly. But here what has happened is, here instead of the sister chromatids equally separating to ultimately give 2 chromosomes in each cell, what happens is one of the cell is not segregating properly and gets 3, and its concomitant daughter cell gets only 1. But in this case, 2 of the daughter cells have a normal phenotype because non-disjunction has happened only in the meiosis 2 and not in the meiosis 1. So these small mistakes can lead to catastrophic defects because chromosomes essentially contain the genetic material. It contains instructions for synthesizing RNA and protein in the cell. So if there is excess of something, there is more protein being synthesized, the instructions are all wrong. And if, at the same time, if there is less of a chromosome, then again, there is not enough protein, because of which there is not enough instruction. So again, let us look at meiosis 1 in order. So what happens is, there are two, say this cell has only 2 chromosomes and they have a sister chromatid each because DNA replication has happened. Now in mitosis, we have seen that upon alignment at the center of the cell, each sister chromatid is attached to 2 opposite poles. So that is what happens is, when the pull force is generated, both the sister chromatids separate, one goes to one daughter cell and the other goes to other daughter cell. But in meiosis 1, this is different because what happens is both the sister chromatids are attached to the same spindle. So here we can see clearly that if the red is one set of chromosomes, the gray is the other set of chromosomes, and both of them have sister chromatids along with it. Upon meiosis 1, both of these are attached to the same spindle pool, and when segregation happens, the two homologs, what are homologs? They are the two copies of the same gene. So essentially, when fusion happens, the same chromosome is inherited from the maternal gene and from the paternal gene. So we have a diploid state in our cells, which means that we have two copies of the same chromosome, one inherited from the mother, one inherited from the father. In meiosis 1 itself, there is a key event that happens, which is known as homologous recombination, wherein these two copies align close together and exchange genetic material between them. And that is what results in genetic diversity of the offspring. But when this happens in this meiosis 1, both the sister chromatids are segregated properly to different to the same cell instead of different cells. And ultimately in meiosis 2, it is very similar to mitosis, wherein the same sister chromatids, they are attached to two different poles and they are pulled apart during cell division. So this is another dramatic representation of failed meiosis. Earlier in the representation, we saw that one cell had a chromosome less. However, in this dramatic representation, what they have shown is if the cell has two chromosomes initially, because of non-disjunction, one cell has both the chromosomes, whereas the other cell has no chromosomes at all. This results in two cells having no DNA at all, and that is lethal. The cell have no instructions and do not survive at all. Similarly, if non-disjunction happens in the second meiosis as well, instead of separating into two different cells, both are going to the same cell, and one cell results in no chromosome at all. This is a dramatic representation in order to understand the gravity of this problem, and often they are manifested in various clinical conditions, and it's often because of aneuploidy rather than polyploidy. So when these chromosomal segregation problems happen, they can either be polyploid or aneuploid. So what the difference between the two is, if an entire set of chromosome is extra in a cell, it is polyploid. So here in this representation, there are 4 different chromosomes. A normal cell has a diploid condition, which means every chromosome, there are 2 copies present. Now say every chromosome, there is an additional copy present, which is represented here. This is polyploid because every chromosome of the cell has an additional copy, resulting in 3 copies in the cell, making it a triploid state. Again, if there are 2 additional copies, making 4 copies of each chromosome in the cell, it is a tetraploid. These are often common in plant cells, but have catastrophic defects in animal cells. In aneuploidy, what happens is there is an addition or a depletion of one chromosome in the entire set of chromosomes. So we can see here that only in chromosome number 2, there are 3 copies. Remaining all chromosomes are normal. This is called trisomy. And because it's in chromosome 2, it's called trisomy 2. Similarly, here, because it's chromosome 1 is depleted of a copy, it's called monosomy. Again, because it's depleted in chromosome 1, it's called monosomy 1. We have all often heard of diseases like Down syndrome, which is trisomy 21, or Turner syndrome, in which patients lack an X chromosome. And for this to be studied, we actually need to know which chromosome there is a defect in. And for that, actually, there is a technique known as karyotyping, and we will look briefly into that. So human chromosomes present in the cell are present as pairs. So we are a diploid cell. There are total of 23 chromosomes, and the final 23rd chromosome is called the sex chromosome, which decides whether you are a biological female or a biological male. If you have XX chromosome, you're termed as a biological female. If you have an XY chromosome, you're termed as a biological male. The remaining chromosomes are autosomes and often are present in all the cells, and they play other functions like developmental or other traits apart from determining the gender. So if you actually think about it, the chromosomes in the cell are all scattered. If you have 23 pairs of chromosomes, you have 46 chromosomes in total, and they all have a huge amount of genetic material. So if you actually just look at them under a microscopy, you will not be able to differentiate what is chromosome 1 and what is chromosome 2. For that, there is a technique known as karyotyping, which basically is pairing and ordering all the chromosomes of an organism, essentially generating a genome-wide snapshot of your genetic material. So for this technique to be performed, what they do is they arrest the cells in a mitotic phase. So they first stimulate the cells with mitogens, induce them to divide, and then they arrest them in the mitotic phase using drugs. So why mitotic phase is because the chromosomes in this phase we've seen are condensed. That is what you want to visualize. Otherwise, the chromatin is just a long thread-like structure, which will not really give you a picture of what is going on in the cell. But even though you do this, and even though you get a picture of all the chromosomes present in the cell, the structural details and what chromosome is what, where the abnormality is, where there is a defect, is very difficult to be detected by light microscopy. So for this, you need to stain the chromosome somehow, and not just stain it equally. You need to stain it in a way that it is being able to be differentiated from each other, that it has some unique characteristic to every chromosome. And that technique is known as the banding technique or karyotyping. So what they do is you take a cell sample. The cell sample can be taken from anywhere. It's typically taken from a cancer cell to see what the genetic abnormalities are present within the cancerous cell, or from the amniotic fluid to see if the fetus has developed any genetic malfunctions or genetic diseases, which would later on lead to a miscarriage or a stillbirth. Then you add the mitogen to the cells. So this induces cell division, induces mitosis, and then you add this drug known as colchicine. Colchicine arrests all these mitotic cells in the metaphase, and the cells are unable to progress further. Then you add these cells to a hypotonic solution. This lyses the cells and releases out the chromosomes. Then you add any fixator in order to fix the chromosomes onto the cells. So by this, what happens is it is unable to be washed out from any staining. And then you dry these slides. You stain them with a stain known as, in here we have represented this using a Giemsa stain, but there are different banding techniques for which different stainings have been used. Then you observe these cells under a microscope. Finally, after observing them and photographing each of the different chromosomes, they can be arranged in an order from chromosome 1 to chromosome 23, and that final image that you get is known as a karyotype. So like I mentioned, there are different types of banding. It's based on what question you want to address, and we'll go one by one. And the first banding that was performed was known as the Q banding. Here, after fixing the cells and drying the slides, the chromosomes are stained with this fluorescent dye known as quinacrine. And what this basically does is it alkylates the DNA and in turn stains them. And it's able to stain the AT-rich region more easily than the GC-rich region. This is because we've seen that the AT-rich region has only two hydrogen bonds between them, but the GC bases have a 3 hydrogen bonds between them, because of which it is relatively easier to access the AT-rich region. They're relatively open, but the GC-rich regions are very condensed, and they are often not stained. So brightly fluorescing regions implicate those regions that are rich in A and T. So we can see here that every chromosome has a unique banding pattern. This is because these regions have these AT-rich regions, and this is conserved across a species. As we know, most of our genetic material is shared, almost 99% is identical amongst individuals of a species, and there is a very small region which is different and which is what gives us the genetic diversity. So overall, when we are staining these chromosomes, they all have these characteristic patterns by which we are able to differentiate what is the first chromosome, what is the 10th chromosome, and so on. So even if it is a mess like this is essentially how you take the picture, the representation on the right. By looking at these chromosomes and their banding patterns, scientists are able to assign them a number and arrange them by order. The second type of karyotyping, this is more common nowadays because the Q banding, what happens is it's fluorescent, so you cannot view it under bright microscopy. You need a fluorescent microscopy to visualize it. And also the stain, the dye quenches with time. So it's a very short-lived image that you get. And as and when you delay the imaging, the signal will get weaker and weaker. So as an alternative, researchers developed this G banding technique in which the GIMSA dye is used. Here, the cells can be visualized by a normal bright field microscope itself, and it is stable and can be visualized over a long period of time. This also has a very similar principle. The heterochromatic regions that are rich with A and T are stained, and the ones that are euchromatin, which is rich in G and C, are not stained as brightly as that with A and T. So again, there is a banding pattern. AT regions are brighter and GC regions are lighter, because of which there is again a characteristic pattern obtained. Based on a similar technique, but the processing of the sample is slightly different, scientists have also developed R banding technique. In this, what they do is before staining the chromosomes with the dye, they heat it to 87 degrees. Here, all the AT region, which as I said is relatively more open in nature, they are denatured because it's easier to melt them. The melting temperature is much lower than the GC region. They are melted and denatured completely. So they do not take up the stain anymore. However, the GC region, because it's a relatively more stable structure, it still is capable of taking up the stain. So R banding, essentially termed from reverse banding, is named so because if you see in G banding, like I mentioned, AT regions are darker, but in R banding, GC regions are darker. So it's a reverse of what you get from the G banding. Now, this technique, why was it developed? Because the telomeric ends that we have seen in previous sessions, if you remember, are regions of DNA that are present towards the end of the chromosome and mainly present only because you have this end replication problem. Your DNA polymerase is unable to synthesize a DNA strand de novo and hence require the presence of RNA primers. These RNA primers towards the are synthesized from the 5' to 3' end, and hence there is a small fragment of DNA at the end of every replicative cycle that is not being replicated. That is known as the end replication problem. So in order to avoid loss of essential genes, the cells have developed presence of a telomere, which is basically repetitive sequences of DNA that are present towards the end of the chromosome that keeps getting shortened and shortened, which with every replicative cycle. So in R banding, because those regions are more uh, they are less AT rich, those regions take up the stain. But however, in G banding, those regions do not take up the stain. So here you can see in the image itself, the telomeric ends are more brighter. They have not taken up the stain. But in R banding, the telomeric ends are more visible. They're more stained. This is a similar banding technique that is also being used with another dye, such as acridine orange, which also stains the DNA on a similar pattern. This is known as T banding. Here also, this is specifically designed to stain the telomeric ends of the DNA. Here also the samples are being heated, AT regions are denatured, and GC regions take up the stain. Another type of banding that the researchers have developed is C banding, which is also using GIMSA dye only. But here there is a series of chemical treatment to the chromosomes after which it is stained. So first there is a treatment with an acid. Here all the purines are removed. So the nitrogenous bases are of two types, purines and pyrimidines. If your nitrogenous base is a purine, it is removed. and then it's followed by an alkaline treatment, barium hydroxide. This reduces the aporenated sugars, and finally, treatment with hot salt solution will result in a chain breakage in that region. However, if it is towards the center of the chromosome, which is the centromere, which is often known as the constitutive heterochromatin region, here there are a large amount of replicative region, and this resists the chemical treatment because they're very highly condensed and very closely packed with each other. So the chemical treatment is not able to cause much significant damage to this region, and only this takes up the stain. So this is known as C banding if the researchers want to specifically stain the centromeric region, because that is what is crucial to mitotic division. We've seen that that is the region that attaches to the spindle pole. And if they want to test for any genetic deformities in that region, then they use this C banding technique. So, like I said, meiosis can go wrong very easily, resulting in aneuploidy. Aneuploidy meaning there is an excess or a depletion of a particular chromosome in the cell. And if there is an extra copy of any chromosome, then it is known as trisomy. Now, we would have all heard of this trisomy 21 condition, which is a clinical manifestation known as Down syndrome. Here, the patient has an extra copy of the 21st chromosome. And this again is a karyotype, and this is very important for the identification and diagnosis of such genetic malformations. So, how can trisomy 21 occur? Say this is an egg cell and this is a sperm cell, both are the gametes that are yet to be formed. They have 46 chromosomes each. After meiosis, they should ideally have 23 and 23 chromosomes. Now, considering there is a non-disjunction, which means that there is improper splicing in daughter cells, what happens is there are, instead of 23 and 23, there might be 24 and 22. While 22 chromosome is very less and is genetically lethal, the one with 24 chromosomes survives, is capable of fusing with the other gamete, resulting in a zygote that has 47 chromosomes instead of 46. Now, this ultimately results in daughter cells after daughter cells, making your entire body filled with cells that have 47 chromosomes instead of 46. This trisomy condition, like I mentioned, results in Down syndrome, which has various symptoms in a child, including mental retardation and a characteristic protruding tongue or congenital heart defects. They, even though their lifespan is not affected, they have multiple other problems that they'll have to face just because of the presence of an extra chromosome. Similarly, there is also a syndrome known as triple X syndrome, in which there is a presence of an extra X chromosome. These patients biologically female, but they have an extra X chromosomes present, and they're also having a large number of symptoms, including again heart defects and underdeveloped secondary sexual characters and underdeveloped ovaries. This again is because of non-disjunction only. Here there are two representative images. One, a problem with the proper meiosis occurring in the mother, and other is the improper meiosis occurring in the father. So let's look at both these conditions. So we know that this is the father cell because it has one X chromosome and one Y chromosome. In meiosis 1, what happens is it's divided. One cell gets the X chromosome, and the other cell gets the Y chromosome. These sister chromatids are then divided into two daughter cells, ultimately resulting in 4 haploid gametes. Similarly, in the mother, there are two X chromosomes. Both are divided into two daughter cells in meiosis 1, and in meiosis 2, what should ideally happen is the sister chromatids separate and there are 4 haploid gametes formed. But if there is a non-disjunction in this stage, instead of it getting separated, one cell gets both the copies, and by chance, if that gamete is fusing with a normal male gamete, then the daughter cell formed will then have 3 X chromosomes instead of 2. Again, if there is a problem with non-disjunction in the paternal gamete, again, the X chromosome present in one cell after meiosis 1, instead of segregation into 2 haploid cells, it becomes diploid in one cell and there is a null in the other cell. If this gamete by chance fuses with a healthy female gamete, then instead of 2 X chromosomes, there are 3, resulting in triple X syndrome. Now, an opposite of this is the XO syndrome, often known as the Turner syndrome. This is this occurs because of a problem with, again, the proper separation of the X chromosome, and instead of getting 2 copies, here the daughter cell gets only 1 copy. They also have large number of syndromes, including heart defects, improper secondary sexual characters, and underdeveloped ovaries. They often never start menstruating, and there is infertility seen in these patients. So let's look at the same image, same representation that we saw previously. So we have previously seen that if in the maternal cell there is non-disjunction in meiosis 2, with one cell having 2 X chromosomes and the other not having anything, by chance if this null cell is able to fuse with a healthy father, or if the null from the father is able to fuse with a healthy mother, this will result in 1 copy of the X chromosome, and this is what is creating Turner syndrome. But why is 1 chromosome not enough? In fact, if you have heard of this thing known as X inactivation, what happens is in a normal female cell, there is in a normal female cell, there is inactivation of one of the X chromosome, which means that even though the cell has 2 X chromosomes, only one of them is actively participating in protein synthesis. However, at the same time, not entirety of the second chromosome is silenced. Still, a very small amount, somewhere around 10 to 15%, is still active and synthesizing RNA and synthesizing protein. This 15% is what is the reason for the symptoms in Turner patients, and in fact the symptoms for triple X chromosome as well. If you think about it, if there is some 15% of gene that whose function is required, and if it is lacking, of course that function cannot be substituted by any other protein in the cell, and because of which there is some symptoms, some developmental defects. At the same time, if instead of 15%, now there are 2 chromosomes, 15, 15 each, and instead of 15, there is 30%, which means that there is some protein that is overexpressed and again results in clinical manifestation. One such protein that was identified is known as the SHOX gene. This gene is actually a transcription factor and has been proved to be crucial for the normal development and maturation of the skeletal system. This is present both in the X and Y chromosomes. So a healthy cell will have 2 copies of the gene that are being always transcribed and translated. If you have X0, which means that there's only 1 copy per cell, there is a deficiency in the transcription factor. And if there is triple X, there are 3 chromosomes synthesizing this protein. There is an excess of this transcription factor. So in fact, if you do look at the symptoms of Turner or triple X syndrome, there are the short stature or very tall characteristics that's seen in triple X because of the development of the skeletal system. There are, of course, other proteins as well, similar to the SHOX gene, that is responsible for these traits. And you can look into them if you are interested. So we've spoken so much about why meiosis is required, why meiosis has to be accurate. Now let's look at the molecular mechanism that drives meiotic phase. The first is the meiotic S phase. Like all cell division, meiosis also requires DNA replication. So similar to mitosis, here DNA replicates instead of 1 copy of every homologue. There are now 2 copies. Both the sister chromatids are held tightly together by cohesins. And in the chromosome, you have these 4 arms, 2 arms of one copy and 2 arms of the other copy. The cohesins are these giant ring-like structure that surrounds the 2 sister chromatids and holds them together in one complex. This is very essential because otherwise the 2 chromatids can float away from each other.
resulting in non-destruction again. So in meiosis 1, like we saw previously, there needs to be alignment of the homologs. In mitosis, it's alignment of the sister chromatids in the cell equator, but here it's the alignment of the homologs. So both the homologous copy needs to come together. They need to recognize each other. They need to associate together physically with each other. And this is the biorientation, which means that one homolog entirely, both sister chromatids together, attaches to one spindle, and the other homolog, both sister chromatids together, attached to the other spindle pole. So in mitosis, often these homologous chromosomes, they act independently of each other. Even though you have two copies of, say, chromosome number 22, both of them are oriented differently. They act independently. They are segregated independently. However, in meiosis, they come together and they segregate properly into two daughter cells. So how does this happen? This happens in the meiotic prophase of meiosis 1. So this is termed as prophase 1. This pairing of homologous chromosome happens because of a complementary DNA sequences, and these sites are known as pairing sites. There are certain proteins that are present in the meiosis 1 that are absent in meiosis 2 or mitosis, because of which this does not happen in other cell cycle. It's very unique to meiosis 1. These proteins promote the association of the two homologs with each other and they get paired together. And this is a four chromatid structure. This is known as a bivalent structure. Here in the representation of a bivalent structure, we can see that, say, this is the paternal chromosome, this is the maternal chromosome. Both have replicated, both have two sister chromatids, and all these four are arranged together in one complex. This is actually setting the stage for something known as homologous recombination, which is the deliberate exchange of materials between different chromosomes. They are essential because that is what generates the genetic diversity that we have seen to be crucial for, say, evolution. The first step in homologous recombination is a break in both strands of DNA. This is a very bold stroke by the cell, and one protein known as POL11 has been found to be crucial for this process. This is a representation of the bivalent structure. You can see that there are paired homologous chromosomes. This POL11 is a protein that is able to cut both the strands of the DNA. But one thing to be noted is that this happens only in one of the homolog. It happens only in one of the recombining chromosome. Now, after this happens, the protein is still attached to the cut ends. So there is a recruitment of a 5' exonuclease that is again specific to meiosis. This protein is able to chop up the 5' end, ultimately leaving behind these 3' overhangs in one of the two recombining chromosomes. The third step is the strand exchange. This is the most crucial step. This is what results in the exchange of genetic material between the two chromosomes. In fact, to imagine, it's very simple. There are 3' overhangs. Now, these 3' overhangs and the other chromosomes have very similar sequences because in, in, in the essentiality, both of them are copies of the same chromosome. So there is a Recce-like protein, which is a recombinase-like protein that catalyzes strand exchange, in which the strand of one chromosome can come and pair with the strand of the other chromosome. This forms a junction, this X-like junction. This is known as the holiday junction. So after a holiday junction is formed, what happens is, again, there is this gap. DNA polymerase is able to recognize this gap and synthesize DNA and fill in these gaps. So there is a filling of gaps, and ultimately, after this, it can be split into two possibilities. We look at the first possibility represented on the left. After additional DNA synthesis, because there is now a template, and polymerase just looks at the template and synthesizes the complementary strand. There is a second crossover. This is known as a double holidays junction. So this is often very common in meiosis homologous recombination. If there is one holiday junction formed, ultimately down the line, there will be a formation of the other holiday junction as well. After this happens, the DNA strands that are forming this X are just cut, and there's a small space that is formed that is then ligated together by DNA ligase. And now this is the hybrid chromosome. It has recombined its genetic material. What was once in one chromosome now is present in the other, and vice versa. Now, another possibility for this is once a holiday junction is formed, the DNA repair proteins or the cell recognizes that, oh, this is something that is not right, because of which it is resolved early on in the holiday junction formation itself, and instead of recombination, it is religated back into its original form and ultimately results in chromosomes without crossover. What is interesting is most of the crossovers are resolved in this way, that they are not successful, they are resolved back to its original nature itself. Only a few of these chromosomes actually are able to undergo these crossovers and move forward to the next generation. So, another, so if there are holiday junctions forming, say, the same place over and over again, then the same set of genetic material will be transferred to the other cell, right? As in the same place is where the recombination will happen. So to avoid that, what happens is there is branch migration in these holiday junctions, which means that, say, this is the branch point. Now there are certain proteins that are recruited to this point, and they have these ATP hydrolysis property, which means that they are able to break down ATP into ADP, and this energy that is released in this process is used for the branch to move along the chromosome. And instead of, say, this position, now the branch has moved to the middle of the representation, and more ATP is used, and it's moved to the right hand side of the representation. So like this, it can move through 1,000 base pairs of DNA, and this is because it's continuously breaking the uh, branch, and then it's reforming the branch, and so on. This is also very essential in order to understand how there is so much diversity in the recombination that we can see. So how does the chromosome look after recombination? So, like we've discussed over and over again, every cell has one maternal chromosome and one paternal chromosome. This is what is known as a diploid state. If there is no recombination that happens, then each haploid will either inherit the same maternal chromosome or the same paternal chromosome. Now, in order to avoid this, and in order to generate genetic diversity, what happens is there is recombination that occurs. And if it is resolved early on, like we saw in the second possibility, there is only a slight amount of uh, DNA, maybe say one base or two base that has been exchanged. Remaining everything is what it was originally in itself. But if there is a proper crossover, then there is a heteroduplex region, which means that one gene comes from one chromosome and the other gene comes from the other chromosome over a large segment of the chromosome. Now there is, of course, there's mismatch between the two bases, right? Like not all bases will be identical to each other. So DNA repair protein will be recruited to these chromosomes with the heteroduplex region, and it randomly corrects. It will not be able to differentiate between what is maternal and what is paternal, what was the original one. It will just take one as a template and resynthesize the other. So in this random event, one allele is completely lost and the other one is retained, which will later be transferred to the daughter cell. So if there is a diploid cell, and here is a representation of one pair of homologous chromosome after duplication, after homologous replication, recombination, and meiosis, it ultimately results in 4 haploid cells, and all 4 are unique. None of them have the same genetic material when compared to the other. So this meiosis 1, the stages that we just described, as in first alignment happens and recombination happens, these have been divided into 5 different stages. It basically starts with leptotene, zygote, pachytene, and diplotin. Ultimately, the last stage is known as dikinesis. That has what is being represented in this image. We'll go one by one. So there is a paternal chromosome, there is a maternal chromosome. They have replicated, so they have sister chromatids each. So each chromosome will have two sister chromatids, so which has been labeled 1, 2, 3, 4 over here. First, in the interphase, they are present as chromatids that is randomly present in a nucleosome like complex. And in order for cell division to happen, it needs to get condensed to form this chromosome that is then separated. So in leptotin, what happens is the chromosomes get condensed and homologous chromosome starts recognizing each other, and slowly genetic recombination is being set up. Then in zygotine, the complex is being formed. The homologs are very closely associated and recombination events are starting to happen. Cachetin is a region where the complex has been formed. The homologs have come to the synapse state, and this is ideal. This is the setup for recombination. The two chromosomes are present very close to each other that the strands can now interchange. In the next stage, diplotin, there is a disassembly starting of the chromosomes. And finally, in diakinesis, the condensation and this condensation of chromosome is lost, and it again starts going back to the division stage. Cytokinesis happens, and then it's getting ready for meiosis 2 to start. So, if you look at this, this is a representative image of homologous recombination happening. Here we can see that there are three events that have happened. One, chromosome 1 has exchanged with 3. So chromosome 1 has exchanged with 3. Chromosome 2 has exchanged with 3. And chromosome 2 has also exchanged with 4. So these events can be captured by fluorescent tags, and here we can see marked by the red arrows are homologous recombination still happening. The two sister chromatids are present very close to each other. But here in D, we can see that this has been complete and they have again disassembled and they are all randomly located. So after this is the actual separation of the two homologs into two different daughter cells. This happens in meiosis 1. So in this, what happens is both the sister kinetochores have to attach to the same spindle pole. And this is what has been avoided, and we've studied how the cell ensures that this does not happen in the mitosis, and how there are so many checkpoints and proteins playing a role in not allowing this to happen. But in meiosis, in fact, this is the very same event that happens. So again, like I mentioned, there are specialized proteins that are present only in meiosis 1 and not in the other types of cell division. So they help fuse the two kinetochores of the sisters together, and they do not help attach to different poles, instead facilitate their attachment to the same spindle pole. Again, because these chromosomes have just undergone crossing over, there is a very strong linkage between them. There is a complex that is being formed. This complex holds the sister chromatids together, and this strong linkage first helps them associate as a pair in the cell equator, and then also helps them separate into two different cells later on in the anaphase 1. Moving on, like we just saw, crossing over is the star of meiosis. It has two major functions. It first, like I mentioned, holds homologs together for alignment in the cell equator, and also, of course, contributes to genetic diversification of the gametes. Now, we've seen that there is this protein known as separase that activates that is activated upon mitosis, and it results in this degradation of protein securin. And securin is what separates the two sister chromatids away from each other. But here, the securin is not destructed and they are held together. In meiosis 2, which is very similar to mitosis that we've earlier seen, the chromosomes align the cell equator. This time, the sister chromatids are attached to two different poles. So when there is a pull of the spindles, they are moving into two different daughter cells. This again happens because of securin destruction. And there is an activation of separase, and the separase destroys securin and it goes to two different cells. So looking at separase and securin again, there is a protein known as separase, which is a protease that is able to degrade the protein that is holding together these two sister chromatids. This is held together by a protein known as securin, which inactivates separase. But in the mitotic phase, there is an active APC complex. This activated APC complex is activated by the CDC20, and this results in the degradation of securin. Now, separase is active and it's able to degrade the cohesin that is holding together the two sister chromatids, ultimately leading to their division. So again, we've seen that the CDC20 comes because there's an increased transcription in the M phase, and also it is able to phosphorylate the APC that the MCDK, which is the CDK cyclin complex, the mitotic phase, is able to phosphorylate APC that enhances this binding and activating the complex. However, in meiosis 1, there is this protein known as sugoshin, which is being derived from a Japanese word that literally means guardian spirit. In meiosis 1, the centromeres are coated by these sugoshin proteins, which is which can recruit a phosphatase. This removes the phosphate from cohesin, and this phosphate is what is recognized by the separase, and separase degrades cohesin. However, because these phosphate groups are removed, separates are not able to degrade cohesin, and in turn remains intact, and sister chromatids cannot separate into two. This sugoshin protein is inactivated in the end of meiosis 1, because of which in meiosis 2 the events happen very similar to mitosis. So with this we'll move on to the previous year questions. So there are 10 questions in total. We'll go one by one. We can try answering it together. They're relatively very simple questions. So the first question is, the diploid chromosome number in an Indian elephant is 56. What will be the chromosome count in an elephant sperm cell? So again, this chromosomes and chromatin numbers are continuously changing throughout meiosis, and let's look at this table that has very beautifully summarized them. So chromosomes and chromatids, we'll look at chromatids first, which is essentially the number of DNA copies or the number of DNA molecules that are present in the entire cell. So in the first prophase of meiosis, DNA replication has just happened. So instead of 23 pairs, there are now double 4n, which is 92, 23 into 4, 92 copies of DNA. Then in the metaphase, it does not change. It's basically just orienting it towards the center of the cell, and it's separating it into two different daughter cells, but still the daughter cells have not replicated. But when cytokinesis happens, this is equally divided into two daughter cells. So there are 46, 46, again coming back to the 2n state. This again undergoes meiosis 2, wherein these 46 chromosomes are all aligned towards the center, and then they are replicated to two different ends. And in the end of meiosis 2, when cytokinesis happens, it comes to n, which is 23. However, if you look at the number of chromosomes in the cell, 46, because both the sister chromatids are present together, and they are present as the same chromosome. So there are 23 pairs of chromosomes, and ultimately the end of meiosis also, 23 pairs get divided into two different cells. So there are only 23 chromosomes now. Again, in meiosis 2, what happens is, in anaphase, the chromosomes are split together into two. So the sister chromatid separates, because of which there are 46 chromosomes that can be visualized by the microscope. And in the end of meiosis 2, when there is cell division happening, it again comes back to 23, and there are only 23 chromosomes present in each daughter cell. This again, in a simplified notion, means that if there is 2n in the initial stage, DNA replicates, there's 4n now. Then it becomes two daughter cell, now again comes back to 2n. But again, when second meiotic division happens, it's divided into four different cells, and it becomes n each. So if the question mentions that if there is a cell that first has 56 chromosome and it is a diploid number, so 2n, say this stage, there are 56 chromosomes. Ultimately, in the last stage, there should be n chromosomes, which is half of this, meaning 28 chromosomes. So the haploid number is a 28 chromosomes. So that is a simple answer to the question. Indian elephant having 56 chromosomes at the end of meiosis, which results in the formation of gametes. So the elephant sperm cell, there will be 28 chromosomes in every cell. The second question, which of the following statements is correct about meiosis? Meiosis begins with a diploid parent cell. Meiosis results in 4 daughter cells, and meiosis is required for sexual reproduction. So, like we saw, again, meiosis starts with a 2n cell, which is a diploid cell, then undergoes DNA replication, and ultimately results in goes through meiosis 1, which results in 2 daughter cells after homologous recombination has occurred, and then finally results in 4 haploid cells. So this is a good time to just summarize meiosis. So meiosis, there is a cell after DNA replication, the homologous chromosomes align together, forming a bivalent structure. Here there is crossing over that is happening, there is strand exchange that is happening, is known as homologous recombination, and at anaphase, the 2 sister chromatids remain intact, but the 2 homologs are pushed towards 2 different daughter cells. These daughter cells again undergo second cycle of division known as meiosis 2. Here the sister chromatids are attached to 2 different poles and they're pulled apart, forming 4 different daughter cells. So again, like we saw, sexual reproduction occurs between 2 haploid gametes. So say we initially had 23 pairs of chromosomes. In a gamete, there are 23 chromosomes each. Now, when they fuse together again, they give rise to 23 pairs in a zygote, and there is an embryo resulting in a baby which has 23 pairs of chromosomes. However, considering that this does not happen, initially there are 23 pairs in male sperm and 23 pairs in female sperm if meiosis does not happen. So the zygote formed and the baby that is ultimately formed will have double the amount of chromosome content when compared to their parents. So it will now have 46 pairs. Now in the next generation, it will double again, 92 pairs, and then it will double again. And this will lead to such a large amount of genetic material that the cell will not understand what to do and will not be able to organize it properly, ultimately resulting in lethality. So, in fact, we've heard of trisomy 21, trisomy at X chromosome, trisomy at 18, or trisomy at 30. These are different clinical manifestations, some of which we have discussed previously. What about trisomy at other chromosomes? Do they always have accurate meiosis? Do they never have any problems? This is not the case. Abnormalities have been detected in almost every chromosome. Like every chromosome can have non-disjunction happening. However, most are embryonically lethal. They result in spontaneous miscarriages and stillbirths, because of which they are not really having any clinical manifestations. Even trisomy 13 and trisomy at 18 have such severe effects that the median survival of the baby born is just 1 year. Trisomy 21 and trisomy at X does not affect lifespan as much, and the patients are often long living. So the correct answer to this question is yes, indeed, it begins with a diploid cell and results in 4 daughter cells, and it is absolutely required for sexual reproduction. So the answer to this question is all of the above. Moving on to the next question. Meiosis 1 leads to the formation of how many daughter cells? So we need to be careful with such questions because it's not meiosis in general, meiosis 1 very particularly. So this is a very beautiful electron microscopy image wherein we can see in the first prophase, they are still present as chromatids, and then slowly in the late prophase 1, is uh, it is condensing to form chromosomes. And here we can see that the sister chromatids are together and homologous chromosomes are being associated with each other, ultimately resulting in, say, strand exchange. Then there is metaphase forming, wherein again we can very clearly visualize that the sister chromatids are being transported together to one daughter cell, and ultimately resulting in two daughter cells. So meiosis 1, which is the first cycle of meiotic division, results in the formation of two daughter cells. The next question was recombination during meiosis leads to what? So we have earlier, I'm sure all of us would have studied Mendelian genetics. Mendelian genetics initially proposed that there are two alleles of the same gene present in every cell. Alleles meaning again copies of the same chromosome that we are talking about, the diploid condition. So say there is a parent with the dominant allele, say RR in this case, resulting in a red flower. And then there is a parent with recessive allele. So both the alleles that are present in the cell have small r, both are recessive and results in a white flower. In the progeny, there is one parent uh, capital R dominant and one parent small r recessive. And in this case, all progenies are red, but their genetic material has one from one parent and the other from the other parent. So in turn, when this F1 generation is allowed to mate together, there is again one inheritance from one parent and the other inheritance from the other parent. This time in the progeny, you can see that there are four different types of genetic material that can be present. And there are different phenotypes also that can be visualized. In fact, there is presence of a recessive type as well, which was hidden in the first generation. However, this is a very simple model. Here there are just red and white flowers. But that is not the case with nature. In humans also, if you take, say, eye color, there are brown eyes, blue eyes, green eyes. There is often blue parents and green parents resulting in brown eyes, or sometimes hazel eyes, or sometimes mosaic eyes. And this cannot just happen if the chromosomes are inherited as such. So this is where recombination comes into picture. And that is what was later added on to Mendelian genetics, which is recombination. This was beautifully studied by in Drosophila models. So say there is a wild type Drosophila. It has a heterozygous gene, and it has capital B and VG, VG meaning vestigial, small VG meaning the phenotype is reflected. So the flies are smaller in size and not developed to its complete size. For the capital VG, the flies are normal. Capital B, the flies have this beautiful color, but if it's both small, then it is a black color fly that develops. So technically speaking, if just Mendelian genetics operated on this, one fly will have capital B, capital VG plus small vg, resulting in the wild type only. The other fly will have small b vg and another small b vg, resulting in a black vestigial fly only. But that is not the case. What happens is there is recombination happening amongst the chromosomes of the parent. So here, the gametes that are formed are not simply capital B and capital B. Of course, that is also present. Small b is also present, but there is recombination happening. Small b is associating with capital B, big B is associating with small b. So these two are the recombinant genotypes. Here, the recombinant genotype cannot arise because they are homozygous. So even if they do recombine, then there will be no difference in the alleles formed. So when these flies are again allowed to mate, now there are very interesting uh, uh, progeny that are formed. The first one will be black vestigial, again a parental phenotype only. The second will be a wild type because again there is one intact dominant allele present. It will be the parental phenotype only. However, the third and fourth phenotype, one will have the big B but will lack the normal VG gene. Hence, it will be gray in color and vestigial, as in the gray color of the parent only, but vestigial. The other one will have two small b's, so it will be black in color, will not inherit the gray phenotype. But because it has a capital V gene, it will have a normal growth size. So these result in the recombinant phenotypes. Even though the percentage of the recombinant phenotypes is very small when compared to that of the parental, it still exists and it's very much present. And this is what gives the genetic diversity that is seen in nature. So again, recombination in meiosis leads to a lot of advantages. One, it forms four genetically unique daughter cells. If you see here, all the four haploid gametes are different from each other. Neither of them are identical to each other. This is because each sister chromatid, the inner ones have undergone recombination while the outer ones have retained its genetic material. It results in genetic diversity and it results in new combination of alleles. The capital B got a chance to recombine with the small vg and the small b got a chance to recombine with the capital G. The next question is, which of the following is incorrect about gametes? So again, we have seen that gametes are a product of meiotic division. What happens is there is a normal cell, say 2n. There is first round of meiosis resulting in two daughter cells. This again undergoes another round of meiosis resulting in four haploid gametes. They are the organism's reproductive cells, and this is what fuses with the gamete of another organism, ultimately resulting in formation of a daughter cell. So the answer to this would be that gametes are organisms' reproductive cell. Gametes are haploid cells. Male and female gametes are known as sperm and ova respectively. All three are correct. But the statement in between says that the gametes are produced by mitosis. We know by this week's course content that this is not true. It is produced by meiosis because it needs to be a haploid cell instead of diploid. An interesting thing that is often seen in the production of gametes in humans is that in the meiotic division of oocytes, the two daughter cells formed are not identical. One is highly polarized, one has a large cytoplasm, and the other cell that is formed is released out as a polar body. This is again um, also true for the second meiotic division. The second meiotic division also results in a polarized cell, and the daughter cell released is a very small polar body. This type of cell division is known as the asymmetric cell division. So symmetric cell division will result in two identical daughter cells. That's what we've discussed over and over again. The genetic material is the same, the cytoplasmic content is the same, so the daughter cells are identical in nature and they have the same traits. However, in certain conditions, this cannot be followed. You need to have asymmetric cell division. For example, in the case of oocytes, this is essential because an oocyte has a lot of nutrients, lot of cytoplasmic contents that is required for embryonic division and embryo formation and later the development of an organism. In fact, you can see that the sperm of such organisms are often highly reduced. They have very little cytoplasmic content and do not have much nutrients to supply to the daughter cells that are formed after fertilization. Hence, it is very essential for the retention of the maternal components. And this is ensured by this asymmetric cell division. So what happens is actually very interesting. There is a spindle migration that happens. So in both the representation and the fluorescence staining, we can see very clearly that first the meiotic spindle is forming again correctly at the cell equator only. It's formed in the center of the cytoplasm only. But later, what happens is there is a movement of this spindle to one pole. So normally what happens is in these dividing cells, there is spindle elongation, anaphase occurs, chromosomes are pulled towards two ends of the cell. But instead, in asymmetric cell division of oocytes, the entire spindle complex moves to one end. These oocytes often do not have centrosomes, so it has these other mitotic microtubules organizing centers, MTOCs, that we have discussed in previous discussion sessions. So the entire spindle complex moves towards one end of the cell. And then there is an actin cap that is formed. So this actin cap pinches out the cell because it forms a contractile ring and pinches out a small polar body that completely lacks any genetic material. Entire genetic material is retained within the uh, mother, and one copy is lost in the small polar body. So this, say, is the actin, right? The red ring. First, it's present throughout the cell, and then we've seen that it kind of has to form in the center of the equator so that it pinches out a contractile ring and forms two equal daughter cells. But in this case, after spindle migration happens, we can see that the actin gets concentrated towards one end only. So there is an actin cap that is forming concentrated actin, and then it forms a contractile ring, and it separates out. It pinches out this very small polar body. So this happens in the second cycle of meiosis as well. So essentially, not much of cytoplasm is given out to the daughter cells, and they just remain attached to the oocyte in the end, as we can see here. The two polar bodies just remain attached. And the sixth question, moving on, is which of the following best defines zygote? So we have seen previously that the meiosis 1 occurs through 5 different stages that have been termed differently based on what happens in these stages. So it starts with leptotin. This is where chromosomes get condensed, the formation of complex is slowly starting, and the homologous chromosomes are trying to recognize each other and uh, trying to come together. Zygote again is where the chromosomes are now recognizing each other and they're getting paired, and the synaptic complex is going to start. It in fact starts in the late zygote and moves on and becomes completely formed in the pachytene, where the strand exchange happens. So zygote is the stage where the synaptic complex starts forming. It's where the synapse begins to get formed and a tetrad formation starts to happen. So, which of the following best defines zygote is a pairing of homologous chromosomes, because tetrad formation gets completed in the pachytene, crossing over happens much later, and termination happens in the diplotin, which is a much later stage. So, in zygote, the thing that happens is the recognition of homologous chromosomes with each other. They start getting paired and they slowly start forming the complex. At which stage does the synapse complex disassembly occur? Again, looking at the same representative image, we can see that the termination of the synapse complex happens in the diplotin. Here, the strand exchange has happened, homologous recombination is complete, and now the chromosomes have started to again separate from each other so that they can move towards two different daughter cells. So, diplotin is where synapsis ends, and the bivalent chromosome with the chiasma, which is exchanged chromosomal ends or chromosomal material, can be visualized. So at which stage does the complex disassembly occur will be diplotin, which is towards the end of meiosis 1 prophase. So the next question was, in budding yeast, IME2 acts as what? So again, meiosis is also containing a lot of regulatory proteins, just like mitosis. It needs to be very well regulated because it can go wrong very soon. In yeast, haploid gamete formation happens when there is nutritional stress. It forms this spores, which are enclosed within these capsules that are highly protected from environmental stress. So when there is nutritional stress, there is an activation of this compound called IME1, which further goes and activates IME2, which is the candidate in question. IME2 is actually a kinase which is able to add phosphate groups to various proteins that initiates meiosis and ultimately starts meiosis. So IME2 is only active in meiosis. So it's a meiosis specific gene, and its function is to phosphorylate. So it's a kinase. So the answer to this question is fairly straightforward. It's a meiosis specific protein. It's a meiosis specific kinase. Moving on, which of the following is a transverse filament protein present in the synaptonemal complex? So we've seen and spoken about this complex synapse complex it forms. And what essentially happens is, say, that there are chromatins, two different chromatins that have to be associated with each other because they're homologous in nature. They're condensed to form chromosomes. Now, there are these lateral elements that are comprised of a large number of proteins, including REC-like proteins and SMC-like proteins, which we have seen previously to be a cohesin complex. So, a large amount of cohesin complex associates laterally along these chromosomes and form the lateral elements. Now, when the two homologous chromosomes recognize each other, there are these transverse filaments that are formed that connect one lateral element to the other, and then they are pulled together, forming very close synaptic junction, which sets the stage up for homologous recombination. These transverse filaments are formed by a large number of proteins. What is characteristic to these proteins is that they have these coil domains. So these coil domains can interact with each other and stack over each other and pull each other together towards each other. In the yeast, these filaments are made up of these protein known as ZIP1, which what was the question? Which of the following is a transfilamental protein? So ZIP1 is the protein. The final question was in RAN-GTP complex, hydrolysis of GTP to GDP occurs during what? So this is a question that talks about nuclear import and export. So let's look at what essentially is this nuclear import and export and how it's regulated in the cell. So we have seen that the cell has a nucleus. The cell, all eukaryotic cells have nucleus, and these nucleus are surrounded by a membrane known as the nuclear membrane. Now, there are pores in the nuclear membrane known as the nuclear pore complex, and they are essentially basket-like structures that form these pores on the double membrane of the nucleus, allowing transport from the cytosol to the nucleus and from the nucleus to the cytosol. However, this needs to be very well regulated, because what happens is, again, like if we have to imagine, let's think about how our RBCs and our liver, or say our heart cells and our brain cells, have the same genetic material. However, they are extremely different from each other. They do not even perform the same function. They do not have the same proteins. They do not even look alike. How? How is this achieved? This is because there are certain regions in, say, a heart that is silenced. Those proteins are not being formed. The same proteins are being formed in the neurons, brain cells. Similarly, there are certain proteins that are silenced in the brain cells that are being formed in the heart. How is this regulated? While there are a large number of regulation and a very large complex, there are kinases just present one below the other. One has to phosphorylate the other, and it goes on. There are cascades. There are signals that are responsible. But one very fundamental regulation that the cell follows is the localization of certain proteins. So say there is a transcription factor that needs to produce a particular protein A. That protein, as long as the transcription factor is present in the nucleus, will be synthesized into an RNA, and that RNA will be translated into a protein. But say for some reason that cell wants to shut down the production of protein A. What it just simply needs to do is, instead of reorganizing the chromatin or whatever, it only needs to send this transcription factor away from the nucleus. So now the transcription factor has no access to the DNA, and hence transcription will not happen, translation will not happen, the protein will not be produced. So this is the localization that plays a role in this process, and that is mediated by this family of RAN GTPases. What it basically has is this cycling form of RAN-GTP and RAN-GDP. RAN-GDP is present excessively in the cytosol. RAN-GTP is present excessively in the nucleus. So this creates a gradient. Whenever there's RAN-GTP present, it tends to move towards the cytosol because there is lower concentration there. And whenever RAN-GDP is present, it tends to move towards the nucleus because there is a lower concentration there. But again, there are these two other proteins, RAN-GAP and RAN-GEF. What it does is, as soon as RAN GTP moves down the concentration gradient into the cytosol, this RAN gap removes the phosphate group and makes RAN GTP. Similarly, as soon as the RAN GDP moves into the nucleus, there is this RAN GEF, which adds or which removes the GDP and adds the GTP on, and again maintains this gradient sort of. So there is continuous cycling of these RAN GDP and GTP from the nucleus to the cytosol, from cytosol to the nucleus. This is what is exploited by these nuclear import and export proteins. So let's look at how nuclear import happens. Say there is a transcription factor that needs to be transported into the nucleus. So there is a cargo. It has a nuclear localization signal, which means that the cell knows that this needs to be transported into the nucleus. There is an import receptor that specifically recognizes this complex and binds to this nuclear localization signal. Now, once this is bound, the import receptor and the uh, cargo, it moves into the nucleus. Here, what happens is the nuclear import receptor is recognized by RAN GTP, and quickly RAN GTP binds to this import receptor. This binding results in a conformational change that releases the cargo. So now, very elegantly, the cargo has been delivered into the nucleus. At the same time, now the RAN GTP following the concentration gradient has to move into the cytosol, and the input receptor is again transported into the cytosol, where it is ready to bind to other cargoes and continue this cycle. Now, the in the case of nuclear export, say there is some protein that needs to be pushed out of the nucleus. There are export receptors that come into picture. So these export receptor here in the nucleus binds the cargo and the RAN GTP together. This moves into the cytosol because RAN GTP now has to move into the cytosol along the concentration gradient. But as soon as it is going to the cytosol, it is converted into RAN GDP. Because of this, again, there is a conformational change in the receptor, and the cargo again very elegantly is delivered to the cytosol now. This empty nuclear export receptor again moves into the nucleus to bind to fresh uh, cargoes to transport them back to the cytosol. So the question was, once we know this, it's simple. In that GTP complex, hydrolysis of GTP to GDP occurs where it happens in the nuclear export in the cytoplasm. This triggers the release of the cargo into the cytoplasm. So that is it. With this, I have ended my presentation. So if we have any questions with this week of the course content, we can discuss. Is there any questions? Is there something that we need to discuss about? Okay then. Thank you all for attending this session. We will meet again on Monday next week from 6 to 8. If you have any doubts, you can discuss with me then, or send me a message in the discussion forum. Thank you. Have a good day. I will be ending this meeting.
Thank you, ma'am.